Hello, everyone. Welcome to tonight's Escarpment Labs webinar, continuation of our webinar series, trying to get some useful educational information and just some conversation going with the craft beer and home brewing communities. Today we have Jonah Greenbaum-Shinder from our team. Jonah's homebrew lead. He's also been pretty heavily involved in some of the projects that we do, isolating and testing out wild yeasts from different places. And then he's also been spending some time at home in the last couple months. And it's been a couple months. That's crazy. He's been spending some time testing out some other food cultures like koji and tempeh, and exploring what we can do with some of the other microbes that are out there that can create cool flavors in food. I do want to also mention we have another interview style webinar this Thursday. That's with Lager Dave Sainer from Elora Brewing Company. Bit of a local legend, this guy. Always love talking to him about beer. He's got a great perspective, a lot of focus. Going to be great to hear what Dave and Nate get up to talking about when it comes to making lagers. And then next Tuesday, we have a special guest, Alexis Wagner from Canadian government. She's going to be talking about kombucha production, how brewers can learn how to make kombucha, and some of the pros and cons and opportunities and challenges in kombucha production. And I think this is something a lot of brewers are thinking about is how can we get more value out of the equipment that we've invested in. And we know a lot of people are thinking about things like hard cider or hard seltzer, but also kombucha is another option. And it's a delicious non-alcoholic thing, and in many ways it's easier to make than something like a non-alcoholic beer. So really looking forward to that talk as well. We are going to have another brewer interview next Thursday, just sort of nailing down some of the details on that before we go ahead and announce that. So without further ado, I'm going to give Jonah the floor to tell us about good wild yeast hunting.
All right. Welcome from near and far. Good morning, good afternoon, and good night. Oh, that's bizarre. Bear with me one moment. I apologize. There it is. All right. So good wild yeast hunting. Today we're going to be talking about all things to do with bio-ranching, bio-wrangling, if you will, tracking down yeast from our native areas and trying to make some beer out of it. So apples, of course, being an excellent example of a place that we could find some wild yeast. So firstly, Escarpment Labs, who are we? We're Canada's yeast supplier. We're local. We make liquid yeast and supply to the craft brewing industry. We offer a wide variety of microbiological quality control tests, custom yeast and bacteria development, and strain banking as well. And we're trying to be the best we can at all of these things. Jonah GS, that's me. Before the beer industry took me in, I had a proclivity for performance production in the theater, and I'm currently getting my Master's of Business and Entrepreneurship and Technology at the University of Waterloo. I'm the head of Homebrew at Escarpment Labs. I am generally the person doing the wild yeast wrangling, and I've been exploring koji and tempeh, as Richard mentioned. Current favorite beer styles would probably be saison and dark lager, or any lager. Today it's a pilsner, but tomorrow who knows. And I came to this industry from a love of brewing at home. So yeah. All right, so wild yeast. We're going to talk about yeast genetics a little bit and the flavor diversity that comes with different types of yeast. We're going to cover how we capture and culture wild yeast and how we're screening for these wild yeasts. And then how we assess, and also how you at home or in a brewery might assess wild yeast or wild microbes for fermentation of your beer or cider or wine for that matter. So the background, yeast, as many of you probably know, is the brewery workhorse. We've long, studied and looked at yeast to try and understand how it functions and the genetics and flavors that can come from one yeast versus another yeast. And then thanks to online communities like Milk the Funk and our Homebrewing and the Brewery, there's been a big reemergence of beer fermentation styles with wild yeast, mixed cultures, and Brettanomyces. And I think in general, as we're all moving towards kind of the local, especially COVID era, hyper local ingredients and getting our products as close to home as we can, the look for yeast in our backyard has come up more and more. So why wild yeast? And here we can see a wheel of all the beer yeast families from Britain and Belgium is on there, to wine yeasts and beer 1 and 2, and the sakes and Asian microbes are over there as well. And many of you know, a British ale yeast might have a little bit of butteriness. It's likely going to flock, whereas a Belgian yeast is going to be more phenolic and likely more highly attenuative. There's a whole wide variety. And so where does wild yeast kind of fall into this? Wild yeast is typically going to be on the higher attenuation side of things. We're going to get some phenols. We might have some overripe fruit flavors. And then also we can get into some weirder, sometimes nastier flavors and smells like socks and Band-Aid. We really run the gambit with what is possible when we're capturing wild yeast. So wild yeast versus regular yeast, or like domesticated yeast, I guess you could say. Often we're going to see wild yeast is highly attenuative. Likely it is not flocculating very well, so it doesn't drop out of your beer as quickly as a domesticated yeast would. Often you're going to get a peppery, fruity aroma, and fermentation is going to be kind of middle of the road, as opposed to a domesticated yeast that's not necessarily going to attenuate quite as much, will drop out better. You'll get some fruity esters, but it's likely not going to have that kind of poff aroma that we get from Saison and other kind of Belgian-y yeasts. And the fermentation is probably going to be, at least at the beginning, more reliable. So in terms of where do we find these microbes, the answer is kind of everywhere. For wild yeast isolations that we do at work, I've had samples sent in of apples that we've scoured for yeast, of tree bark. I think a brewery in Ontario sent us 100-year-old fence once. But typically we're like bark, fruit, grass, dirt are all areas where you're going to find wild yeast. If there's a sugar source for yeast to be on, likely in the world, we will find it in these places. So what do we do? How do we find the yeast now that I have a piece of fruit or an apple or some tree bark? So we need to create an environment similar to finished beer in order to suss out whether or not the microbes that we've caught and captured are actually going to be reliable, be able to produce beer and not anything else, or that they are safe at all. So we're looking for something that's hop resistant. We're looking for something that's ethanol tolerant, and we'll discuss how ethanol tolerant as well, and then osmotolerant as well, so that it can handle a high sugar environment like that of wort. So normally in the lab, to get things going, we will take samples as soon as they come in, and we will mix them in with our wort media and about 3% ethanol, so about 3% ABV. And this stems off some of the nastier microbes that might have been kicking around on your apple or orange or tree bark or what have you. And so that's how we begin our process. Normally, we'll leave that there for a week or two. We'll look for signs of fermentation, and then we'll continue on. So once we've selected these samples and we've sat them in at work, we're using Falcon tubes, but a beaker, a beer bottle, anything that would probably do. Then we usually do a series of plating and kind of scale up from there. So we use MRS and WLN plates, which are beer selective agar medias. And what we found at work that is very successful is incubating these plates aerobically in order to inhibit mold growth. Often we'll get plates, if they're done not in an aerobic environment, that you might have some candidates or some colonies on there that within a few days the mold will overtake it and it becomes unusable. So we're isolating in that way to inhibit mold growth. As mentioned before, the wort and ethanol incubation at 3% and sterile, ideally hopped wort, can also help with warding off some of the nastier bacterias. If you're looking for lacto, however, I would probably start with unhopped wort. And then for at home, at the very least, if you want to do successively higher ABV tests with starters, once you've kind of sussed out some candidates, that's not a bad idea, because ultimately we're probably trying to make a beverage that is more than 3%. So we do eventually want to know that we can get to 5 or 10% ABV with the microbes that we're selecting. And then once we get some real candidates, and we'll flesh out how we really determine that as we continue on, often we'll end up doing a homebrew trial. We'll make 20 or 40 liters of wort and then do a bunch of 2-liter or 1-liter fermentations and run sensory on those beers so that we have the control of the wort being the same, and then we can truly evaluate how the yeast is impacting the nature of these beverages. So the old homebrew bottle, it brings me back. So yeah, as mentioned before, apples cut skin off can be great for capturing yeast. Grapes, berries, tree bark. I've also never used but heard very good things about wasps or bees, which are obviously touching all kinds of pollen-y, fruity things throughout their lives. Elderberry I have also not used, and hops is a convenient one as well, kind of in the beer ecosystem already, and people have been quite successful with finding wild yeast there. So if you happen to have an old homebrew bottle, you could do a small little starter in a homebrew bottle and use that as your incubation media and vessel to begin your hunt. So suppose you were doing that. You would seal it and you would watch and wait and see what happens. If you give it a shake in a few days and there's some bubbles, maybe we've got something going on. You could put an airlock on it. That'll also help inhibit mold growth once a bit of CO2 starts kicking up. And then, of course, you have to smell it. The best indicator, at least at the beginning, of something being a potential candidate or being interesting for use in beer is, does it smell good? If it smells like old dirty socks, then maybe we'll move on to the other one. If it smells, I don't know, like an ale, or has some esters or some clove notes that you're liking, then we're going to continue on putting that sample through a few more paces. So then you can step it up to a 1 to 2 liter bottle if you would like, if you've not already done so. And then pH is also pretty important here. You want to get below 4.5 pH for to avoid botulism and other growths that can happen in a more basic environment. So here is, we can see Angus. He's swabbing a apple, which I believe led to a commercialized strain, which we will touch on. So you take 10 to 50 samples out in the world, and then you can see here our falcon tubes with airlocks and the samples within them. So we'll leave that there for a week or two. And then we begin plating. And so this is a plate with some colonies on it. It's probably been run through a few series of isolations in order to get to just one colony. Normally, the first time we plate out from a falcon tube, there's many things. Some look like yeast, some don't. And then once we've isolated down to our previous colonies, we'll select colonies and we'll run PCR, polymerase chain reaction, in order to identify if these microbes are in fact yeast or otherwise, and to see if they have the STA1 gene and are diastatic. Often we'll see wild yeast do have that, but it's not by any means a rule. So here, afterwards, we'll send that to the university to get sequenced. And so here you can see a list of various other microbes that we have come across, not Saccharomyces cerevisiae, that have come up in wild yeast capture. Some of these, the Hansennia spora, we'll find in wine sometimes, or in cider fermentation. Lachantia, I believe, can be used. I might misspeak, but I think it can be used to ferment lactose. And then the Torulaspora delbrueckii, which I've goofed around with a bit as well, isn't a great attenuator, but has smelled really nice. So perhaps could have been used in a mixed culture. So wild capture at escarpment. What we're looking for is we're looking for yeasts that can ferment maltose and maybe maltotriose. Ideally, 5% ethanol tolerance at the very least, but the more the better for it becoming a commercialized strain or being truly viable in a brewery setting. We're not always going to run into breweries that are just making beers under 5 ABV, and even if they were, that's a little dicey. So here you can see some mini test ferments that are being run on strains that have been selected, and then we'll run them through sensory and determine what candidates are really impressive and which ones are the wet old sock of the bunch. So the goal is to find Saccharomyces cerevisiae, but sometimes, as I showed, we come across different microbes that can be used in a mixed culture, or we've come across some Paradoxus strains as well that attenuate well enough that they can be used commercially. So the typical flavors of wild yeast. We're often going to get some fruitiness, but not exclusively. Sometimes there is no fruitiness. Banana is a big one, and the Belgian vibes are also definitely prevalent, at least to the strains that I have smelled. Attenuation for the maltose fermenters in the bunch can be somewhere lousy around 50% to 100%, super highly attenuative, sometimes really fast. It's kind of, no pun intended, the Wild West of looking for strains to make beer with. It is rare, but occasionally we'll come across something in the 75%, not a lot of off flavor or phenolic off flavor vibe, and you can just kind of go about and use it like you would anything else. Typically, it is going to be slower because these yeasts are existing out in the wild world and not workhorse brewery yeasts that have been passed down through generations. So once they pass the test and we've identified them as Saccharomyces cerevisiae, we'll ideally commercialize it and turn it into yeast that people can make tasty beer out of. So here's some examples of some just final information for two of the yeasts that we had previously covered from our U of G case study. So I believe these beers started at 1040. So you can see they're fairly attenuative, and then we've tracked the pH to know that we're in a good zone with pH. Some of the smells and tastes, just to kind of try and help the client to know how to utilize this. If since the gambit is so large in terms of what kind of flavors and performances we can get when we're ranching like this, it's really important to be able to give some clear-cut information and advice to help people along, so you don't end up wasting a batch or a tank or what have you. So here, Wild Thing is a wild capture yeast that is now a commercialized product that we sell. It's decently attenuative. You can see that it performs faster at 75 than 65 Fahrenheit. And then we can see on the right, my right, your left maybe, just the different esters that we're getting and the different flavors that are coming out depending on the temperature. So it seemed that, the lower attenuation at 65, you're going to get a little bit more body, a little bit more linger. 65 might be a little cold for a yeast that originally existed out in the world on an apple, wild strawberry yeast, and this yeast turned into Ontario Farmhouse. A bit of a closer curve, but still fermenting faster at a higher temperature, which checks out. This beer yeast has a bit of a saison-like vibe. I know that it's been used quite successfully in some saisons. I believe there's a Toronto brewer who medaled with Ontario Farmhouse Ale Blend at HomebrewCon last year. So you can really make great products and great beers from these yeasts, as long as you kind of know what direction you're heading with it and how you're trying to use it to craft a better beverage. So just some examples of some breweries that we've worked with that are using wild yeast or cideries in the case of Revel. I mean, Revel is doing a lot of, most of their stuff is not inoculated, so that's a whole other ball game, if you will. But there is viability for these yeasts to become staples in really any brewery, as long as you can find the right use for it. Lots of homebrewers are doing wild capture, like I mentioned, and making their own neat beers from yeast that they found in their backyard, which I think is super cool. I know it's a goal of mine to get some yeast off of my apple tree, and I've got hops off the backyard. So as soon as I start growing barley, I'll be able to make a beer with only ingredients from my backyard. So how can I do this at home? Just as a recap, you're going to want to make about a 1040. That's about 10-ish Plato gravity starter, hopped unless you're looking for lactobacillus. And then you're going to want to add alcohol to create around a 3 ABV tolerance. You can add fruit, bark, or leaves, or you can just leave it in the backyard for a night, probably covered so that bugs, doesn't just get infested with fruit flies or something, and then you just have Acetobacter, and that's not going to be great unless it's a Flanders Red. And even still, you're going to incubate it for a week or two and watch that gravity. Watch for signs of fermentation starting. You can take a gravity sample if you'd like. to track and see where we're at in terms of attenuation. So you want to know, did the gravity drop? Does it smell good? Obviously very important. Did the microbes survive? This is more when you would step it up. You could step it up into another alcohol injected starter, or you could, if you were feeling gutsy, you could put it into another starter without anything and just kind of see how it fares to know if your microbes are still alive. Does it smell good? Does it smell good? And then pH. We want to know if your pH is below 4.5 for health and safety reasons. If so, then you probably have some wild yeast or other microbes that can attenuate beer wort. And why not try and make a beer with it? If you're looking for more information about this, Milk the Funk has a ton of resources on all things weird, wild, and funky when it comes to beer. So I would definitely recommend checking out the group, checking out the wiki. They have a podcast on bioranching as well. That's quite informative as well. And then I guess we're off to questions. So I will stop sharing.
And I'm back.
All right.
Thanks, Jonah. Informative stuff, and I do see a number of questions down here as well. So I think that'll be good to get through and answer those.
Cool.
Provide a little bit of clarity in some of these places. It is a complicated topic, right? Hunting and capturing wild yeast, and there are, I think, as you mentioned, so many different ways that it can be done, and I think even between yeast companies or yeast nerds, it's some people have different approaches, right? I hadn't even considered, for example, the anaerobic incubation of those plates until you started doing it, because my approach was just kind of just gunslinging, get in there, get the yeast before the mold shows up. But yeah, certainly just keeping the air out does make things a lot easier when you're trying to do 4 or 5 big isolation projects at once.
Yeah, it certainly makes it more manageable, and you still run into sometimes a bit of yeast creeping in if maybe the colony hasn't grown up enough and you leave it out for a day after and you want to get out, but it makes it a little bit more peaceful.
For sure, yeah. And yeah, certainly we've seen a lot of success with pulling lactic acid bacteria off of fruit and trees and stuff like that. If that sample goes into unhopped wort, it's you have a good chance of finding something acid producing in those conditions.
Yeah, absolutely.
Okay, so the first question that we have, I'm sort of just going to go in order of how these are voted. Sure. The first question is, when you say hopped wort, how many IBUs are we talking?
I would say 10 or 15, I guess, if you're really not after. We'll run into occasionally, if we're lucky, like a hop tolerant lacto that is fine for 8 IBUs. So I think going over 10, over 15, which isn't really a lot of hops in a starter anyways, that's certainly going to ensure that you're not getting lactic producing bacteria.
Yeah, I would agree. Even the bacteria that have hop resistance, which does sort of jump between bacteria on plasmids, so they can sort of exchange that information. Even so, for most of them that aren't really used to being around hops, 10 IBUs is more than enough to really, really slow them down. And there is a bit of a period of adaptation for even the ones that have that hop resistance adaptation to really get comfortable and quick in that environment. Yeah. We've seen bacteria sour beer at, up to 40, 50 IBU, but, it takes a little bit of boot camp to get them there.
Yeah, totally.
The next question is, does a medium on which yeast grow exert a selective pressure on yeast metabolism? For example, have you noticed interesting properties unique to yeasts growing on certain fruits?
Oh, that's uh, hmm. I have not noticed, but I also have admittedly not really paid too much attention to what the initial sample was. I've kind of honestly been more focused on finding yeast than I have been on what the original inoculum was.
Yeah.
I would look into that though. I think that's really interesting.
Yeah, it's something that we we thought was interesting when we first sort of tested and started testing out the Wild Thing culture with different brewers. Was maybe I think there is an element of psychological priming here because everyone knew that this was a yeast from an apple, but that was always the comment from the brewers and from the people in the tap room was, oh, this really tastes like an apple, really has that characteristic. And it is true in the sense that yeast does produce a lot of those apple esters, right? The fatty acid esters like ethyl decanoate that really does make something smell and taste like apple, but it also could be purely coincidental.
Yeah.
I will say, depending on, the kind of substrate that yeast has been on, that may tell you something about that environment, right? Like, for example, we find that we're more likely to find Saccharomyces, like a certain yeast, like Saccharomyces paradoxus. It's another type of Saccharomyces. We find it on tree bark a lot, and maybe that tells you a lot about how that what that yeast needs in the wild to survive, right? Maybe it's living off of something coming off of those trees, or we associate with certain plants, like we've found a couple times a lactobacillus that's known to be part of the bee gut microflora in flowering plants like thistles, for example.
Mm-hmm. Cool.
I don't know if that provides an answer, but maybe it provides a little bit more context. Here's a good question. What percentage of samples from nature yield a strain that one could make a decent wild ale or saison with?
I think if I think about it, well, you might get more than one potential candidate out of any given sample. So if, let's say, I have a job and there's 12 different pieces of fruit that that we're trying to isolate from, that can explode into 30 or 40 candidates based on the testing that we're running on them. And then 3 of them might be really 1 to 3 of them are going to turn out to be something that you like would think of as a saison maybe, or like have known beer characteristics. It's a low percentage. I feel like it's a lot of work to hunt through them and kind of chop through the numbers in order to dial down to viable candidates. And
for sure, yeah, I would agree. It's very much a numbers game. It's hard to say. I will say, having done a lot of these wild captures, in my mind, like the success rate to get, especially without that isolation step of pulling out individual yeasts and testing them, I would say the success rate is fairly low. But some people have more success than others as well. Maybe that has to do with the environment.
Yeah.
But yeah, I would say, like, if you're trying to look, trying to hunt for a good culture, I would start with 10 samples and sort of go up from there, because it really is a numbers game. I know there's a brewery that I really love in near Philadelphia called Forest in Maine, and every year they essentially forage for that year's saison yeast from plants around the brewery, and I seem to recall they use a couple dozen mason jars to try to hunt for whatever that winner is. Yeah, that's a cool idea, man. Like, refreshing your yeast completely every year as a mixed culture, kind of a good way to keep things fresh. But yeah, like across the board, it seems like in general, this is just you kind of have to try a lot of different things to find that one thing that works.
Mm-hmm. Yeah, I agree.
We have a lot of questions. Wow, this is great. The next question is, what is the best approach for adding alcohol to 3% ABV? So for a 100 ml wort sample, some amount of vodka, for example?
Yes, I would say yes.
Yeah.
I'm always bad at this math, and when you're dealing with something at 40%, it's even more confusing. At the lab, we have 95% ethanol around, so it's a little bit easier. So I would defer to Richard in terms of the numbers of making that work.
Yeah, you gotta think back to your grade 10 chemistry and C1V1, and I think you'll find an answer there.
Yeah, that is it.
That is it. But yeah, I mean, yeah, you can use vodka to bump up the ABV in a wort sample, no problem.
Yeah.
The next question just says cheers. Cheers. The next question is, do you have a printed protocol that you can share?
We don't, but I don't see a reason why we couldn't share something. We do have a blog on the website that kind of details a wild yeast hunt on the University of Guelph campus that might be an interesting place to start, but we could definitely add another blog post with kind of a how-to, some steps and some of the information put up here.
That would be worth doing, I think. Yeah, I think that blog post on our little project at the University of Guelph to capture some wild yeast is a good example. I think we do have a lot of the back end details in there as well.
Yeah,
the more practical thing of like, this is how you make the wort, this is how you set up the jars. I mean, our system is very simple. It's disposable centrifuge tubes with airlocks on them, and that works pretty well. We we forgot about a few in the lab for a few months, and one of them grew a mushroom.
I was embarrassed, and now I'm very, very proud.
I want to find out what that is. I was blown away. I'm like, how the hell did we wild capture a mushroom?
Yeah, we have to put it on potato agar now and see what happens.
I'm gonna get to that this week, actually.
That's sweet.
Next question is, if you don't isolate and brew with a mixed culture after stepping it up in hopped and pre-acidified wort, how diverse is that typically? Does the culture tend to become more dominated by the yeast with repeated use?
I would say that it kind of depends on what microbe or what yeast is in there. I think if you're not going to isolate, then doing like a more intensive starter boot camp would be the way to go, and excessive step ups into higher alcohol, into higher alcohol starters. Is because you want to be able to have a microbe that can sustain a beer environment ultimately. So you're going to negate some of the evil candidates, if you will, just by stepping it up that way. So I think that's probably the best route.
Yeah. Like the thing I would add here is that a lot of mixed cultures do become more simple over time. It's something we've observed in maybe a less than ideally scientific manner, and that we are now working on observing in a much more scientific manner through a project we're working on. Mixed cultures that, maybe it starts with a bunch of different things. After time, typically, you do have a few different characters that start to dominate that environment. And it often does sort of trend toward becoming a little bit more simple, with some exceptions, right? We're still understanding how you can have dozens and dozens of different yeasts in a kvike, for example. But a lot of these things, like, you can also look over at the sourdough world, where you look at a lot of sourdough cultures, and really it's like 1-2 yeasts, 1-2 lactic acid bacteria. It's very simple, but simple communities can also make really complex flavors. What do you think of using grocery store pasteurized apple juice as an initial capture medium?
Sure. I mean, like it's a sugar source. I think if you're not hopping it, then you run the risk of getting lactic acid based things. you're not going to run into the maltose and maltotriose kind of questions, because you have more simple sugars in apple juice. So, I mean, I think I would use that if I was looking for a yeast specifically for cider. Otherwise, I mean, DME is pretty cheap, I would say. So if you're trying to make something or capture something for beer, using beer ingredients makes more sense. But that isn't to say you couldn't capture microbes in pasteurized apple cider or apple juice.
Yeah, the only thing that I would caution about with the pasteurized apple juice is if there's no solids in there, you may not have a whole lot of nitrogen in there. So that means that, especially when you're looking at yeast, which are pretty nitrogen hungry, you might not have the nutrients there for the right yeast to grow. So you may want to spike it with a little bit of yeast nutrient, for example. And yeah, I think yeast nutrient with apple juice would work pretty well for a wild capture. I am a fan of these kinds of off-the-shelf approaches. Like, I will admit that for any kombucha experiments that I've done in the lab, I buy a Tetra Pak of Arizona green tea, and that works great for testing out different kombucha cultures. So I'm all for the off-the-shelf stuff, but I would think about nutrients in the case of the apple juice. In your experience, what source yields the best yeast for you? Flowers, fruit, tree bark, tree sap?
Anecdotally, I would say that I've been pretty impressed with the tree bark samples that I've done isolations off of. Have had diverse and interesting candidates. I have a beer fermenting at work that I'm goofing around with a Paradoxus strain in a mixed culture that was off of some balsam, and it's smelling great. So tree bark has been really cool, and not something when I had initially gotten into doing wild capture that I thought would be as fruitful, if you will.
Yeah. I will agree. Tree bark does give a lot of interesting results. You get a lot of interesting yeasts in there.
You're also going to run into mold in tree bark, I would say.
Yeah.
You want to be careful about. There's going to be mold, and maybe
That's the real reality of anything out in the wild, is there's going to be mold.
Yeah.
They're amazingly competitive.
Yeah.
I will say that, just in my own experience as well, fruit and tree bark have yielded the best results. You often get yeasts from flowers, but it's often not yeast that are very useful in beer. You get all sorts of weird oddball species that don't really show up in fermentation. So not to say that those aren't eventually useful, but maybe not immediately useful for what we're trying to do. But yeah, fruit and tree bark, like apple skins in the fall, almost always you can get something cool out of those. Could I harvest and reuse wild yeast captured from my backyard after fermentation is finished?
Yeah, I mean, I think you could use it like you could harvest it like you would harvest any other yeast post ferment, or if it's happens to be top cropping during ferment. But there's no reason, if it's a viable and the word is escaping me, but stable yeast, then there's no reason that it shouldn't be able to go for a few generations at the very least.
Yeah, for sure. I would say, maybe it might change a little bit over time, but there's there'd be no point just harvesting. Well, I mean, I guess maybe I'm beyond lambic. There's no reason to just harvest yeast once and not reuse it.
Yeah.
You might as well, get more benefit from that effort. If you're on somewhat of a budget, what equipment would you suggest to get started in harvesting wild yeast?
Mason
jar.
Yeah, a mason jar and some I mean, like if you were just trying to do open air capture of some cheesecloth would suffice. Otherwise, a lid. And I mean, an Instant Pot is basically a glorified home autoclave if you're trying to have a bit more sterility. But yeah, you could do it all with mason jars pretty much as far as liquid testing.
Absolutely. Big fans of the Instant Pot and the Anova sous vide over here.
Yeah.
Those two things can even for breweries that are setting up labs, those two things can save you tons and tons of money.
Yeah.
You use PCR for identifying STA1 presence. Is there a good way for us to identify diastatic yeast we collect?
I would say PCR would be the way. That's a bit obnoxious of a response. I apologize. But PCR. STA1 is the gene for diastatic yeast, if I'm speaking about that correctly. So if you have yeasts that are STA1 plus, they have the diastatic gene. Whether or not they exhibit diastatic properties is somewhat different, but PCR is kind of the industry standard, for lack of a better word, for diastaticus identification.
Yeah, I would say, yeah, exactly. That being said, like if you've got a wild yeast and it's super attenuating, like it's just going down to nothing, then there's a good chance that you've got a yeast in there containing STA1. Here's a good question. I don't know if we can answer this. It's a good question. Do you find that dominant wild cultures tend to have a geographic basis? For example, do multiple cultures in the same area tend to have a similar or the same dominant culture, or I guess species, even when captured from different sources in that area?
I'm just rereading this. Do I find that wild cultures? No, I don't think so. I've found like I've gotten multiple candidates off of one sample if we're talking like super close proximity geography, but not necessarily beyond that, I would say. Although in my experience, when samples are sent to the lab, I don't always know, the proximity of one to the other when they're sent in. So I don't have all the information on that. But I have noticed like a tree bark sample that has had two different Saxarvisiae strains on it and no other in a 10 sample trial. So there's nothing there as far as that goes.
Yeah, I think that there's like a little bit of that sort of microecology of, what is found on what things within one area or ecosystem. And, recognizing that most of the work that we do is in northeastern North America, and that itself is somewhat limiting. But then there's also, like the macroecology as well. And if you do look at studies that are done on wild yeast all over the world, there are, some trends. Like, for example, if you're to go into a beach forest in Patagonia, you might find Saccharomyces eubayanus, right? The other lager yeast parent, which it's not so common to find it here. You can find it in northern North America. We've not found eubayanus yet. We've seen it picked up in Quebec and New Brunswick and places like that, but thus far for us, it's eluded us. But for example, here for us, a yeast that we find all the time is Saccharomyces paradoxus, and you don't find that one everywhere in the world. So in some ways, the environment does select for what kind of yeasts or microbes show up, but in the case of yeast and microbes, that environment could be as small as a tree or as big as a continent, right? So it's kind of like this question I sort of gets to like the idea of terroir, and it gets really complicated when you get down to the level of microbes. I remember having a email thread. This was back when I was just getting into microbiology, but with this professor in the US called Ben Wolf. He studies cheese rind communities, and I had sort of naively posed this question of like how how can you prove that cheese has terroir? And, he got back to me with this idea of, making blocks of cheese and shipping them all over the world, and then getting the cheeses back and measuring what microbes show up. And I wish someone had done that experiment. But I think, in the case of just like with lambic breweries, just like the environment of the brewery or the facility itself has such a big impact. It's really hard to prove.
That does make me think of the trial with music being played for cheese to see which was the funkiest, and hip hop was the funkiest cheese.
Yeah, I'm curious about that kind of stuff. We play music in the lab. I don't know if it has an impact on the yeast, but
yeah,
I wonder.
Yeah.
If you play ska, does Cali go faster? I don't know. What wort gravity range do you use for wild capture?
Typically 1040 is as high as I'd like to go. You could do a little lower, but you want to know that it's sugar tolerant. So 1040 is kind of what we're stipulating.
Mm-hmm. And pretty standard gravity. Have you ever brewed with Paradoxus, and was it good?
I have a beer sitting in the lab with Paradoxus, and it smells awesome.
It's a co-ferment?
Yeah, it's a co-ferment with some Brett isolations from a Swedish brewery, I think, and another isolation that I took from another beer. So it is not by any means just that strain in there. But I'm looking forward to packaging that beer. So that's my experience with Paradoxus thus far.
Yeah. There's some interesting stuff. They tend to not attenuate very well, but they do seem to have pretty good biotransformation potential. Like whenever we put them in hopped wort or grow them in hopped wort, you often get all sorts of nice aromas coming out. There was, I think, one that you guys had in that Uwag Wealth project that had a really nice sort of woody, cinnamony kind of character as well. So there's potential. I guess we just really haven't tapped into it yet.
Yeah, we just need to run another trial. Yeah, that beer was Whirlpool hopped with Sabro and smells like Sabro, but it's got some pina colada going on, which I'm excited about. So
yeah. Do you have any way to narrow down what style a yeast might work with, or is it just random testing?
I think ultimately that's going to come down to like, what does it smell like and what does it taste like? Attenuation is going to be obviously a part of that, although you can run into some kind of body deceptions, I would say, with yeast. I know Kvike has done that for sure. Like, taste a beer that is at 10-15 and it tastes a lot drier than that. So there's some trickery there. So yeah, I think you kind of have to follow your nose and taste buds when it comes to what style the final product is going to be best for.
For sure, yeah, and like recognizing that most wild yeasts are going to be phenolic, so that does limit things to sort of the Belgian end of the spectrum usually, but not always.
Yeah, and you'll know. I mean, there's no hiding POF. There's no hiding it. So
no. Extending the geography question, does seasonality make it more difficult to find yeast or certain types of yeast? I realize flowers and fruit aren't an option during the winter.
Yeah, I mean, for at least for like the wild capture program that we run, we try to take samples in the fall. That's often when you'll see breweries cool shipping. The microbes that have survived the summer are, feasting on the fruit and tree bark bounty of the season, and maybe left some of the lesser promising microbes in the dust. So although you can definitely try and do some capturing in the spring or in the summer, the fall is when we've been most successful.
Yeah, absolutely. Just looking at some of the other questions. I mean, it's actually a great question, and not really something we've explored a whole lot. Have you tried harvesting microbes from dirt? I know dirt is full of microbes.
We did get a dirt sample, and one of the ones that grew mushrooms. I don't think I pulled.
That makes sense.
Checks out. I don't think I pulled any beer microbes out of it. Not that I can remember. Nothing that was profound, at least. Which isn't to say that they're there. I just think, there's a lot of biodiversity in dirt. So
a lot, yeah.
Other things might have just been more prevalent and taken over earlier.
Mm-hmm. Yeah, I would say maybe the type of dirt matters. One thing that's known about the Saccharomyces family in general is that they are generally associated with trees, and it's not just the bark where there might be some sap or transpiration that might allow them access to some sugar, but also you have to think about where are fungi the happiest. It's often hanging around the roots of the trees where the trees are also often releasing sugar. Basically, they release sugary exudates from the roots, and that's where you find a lot of fermentative yeasts, because that's just an environment where there just so happens to be anaerobic conditions and also a bunch of sugar. So it's kind of the perfect environment for wild Saccharomyces. So if you have access to, if you're got some oak trees around, for example, you could try digging some soil around that tree and see what pops up there to maybe increase the chances of success.
Yeah,
we haven't really played around with that too much. See, part of it is because just soil just has so much microbial diversity in it a lot of the time.
Mm-hmm.
Just thinking back to second year microbiology class and having to plate out soil and just seeing all sorts of crazy stuff pop up. And probably most of it is not great for beer.
No.
There's a few other questions. A mead question. You've been getting a few mead questions lately. My friend's mom is producing honey and doing mead out of it. 2 of the honey glasses just started fermenting on their own. How would you proceed with them? Adding them to beer wort now or dilute a little bit, adding nutrients and let it grow in that media? Yeah.
Producing and doing mead, 2 of the honey glass.
I mean, it sounds like some water may have got into the honey to allow it to ferment, but I would say if you've got fermentation going on, then just proceed.
Yeah, I would. Yeah, you can step it up into an ethanol starter of some kind, but I know honey is not something I've ever tried to capture off of. But I mean, that's interesting.
Mm-hmm.
Yeah, I would put it into a starter with 3 or 5% if you already have some activity, and kind of see what it's smelling like, and maybe what it's tasting like from there to really put it through its paces. But I mean, it looks like you've captured something already. So
yeah. The only thing I would add just from the microbiology side is honey tends to be actually pretty low in terms of microbe content. It obviously depends on the type of honey quite a lot, but given the environment of just being really high sugar and so really high osmotic stress for microbes, it does kind of create a bit of a selective environment for yeast and actually for lactic acid bacteria as well. So the nice thing about honey is there may not be a lot of yeast there, but there's also probably not a lot of junk there either. So it is reasonably easy to get yeast or lactic acid bacteria out of it. So it is a good place to look. We haven't, maybe we should put a little more energy into getting yeast off honey and honeycombs. Mm-hmm. Honestly, the last time I tried, I had a roommate who's a beekeeper, and went out to his hives and did some swabs, and got stung a few times and kind of just it was a bit sour on that experience. Maybe that's what. Maybe it's my own bias showing through that we haven't really explored this a little further. But bees are great.
Save bees.
Just don't mess with their home.
No. Cool.
All right. I just saw a question pop up. Okay, that's a good question to end on. How often do you find Brett in your wild captures?
I have not found Brett in my wild captures, which is a bummer because I was like, oh, that'd be cool. But no, they have eluded me thus far, which is, fine. There's some of the other microbes have neat interactions with Saccharomyces, and so I'm okay with that. But in my experience, they are hard to pin down.
Yeah, extremely hard to find in the wild. I have not found a truly wild, I have not found Brett from a wild capture, for example. And even more broadly speaking, no one really knows what Brett's natural environment is, other than it shows up in a lot of breweries and wineries, and it's kind of bizarre in that way. It's still a bit of a mystery. We found a lot of things that are similar to Brett, even similar genetically. So yeast like Pichia, which is a lot closer to Brett than it is to Sac, has a lot of similar functions. Like some of those can make the phenolic aromas, like the funk, pellicles, stuff like that. But it's not Brett. I think oftentimes when people report that they've wild captured a Brett, it turns out to be one of these other yeasts, and there's nothing wrong with that. That's great. Pichia is awesome. It's totally the dark horse of the mixed ferm world, and has so many uses, and hopefully we can explore a little bit of that world as well. I use a Pichia in my Saison culture, and it's part of it is just because it produces a pretty gnarly pellicle and looks cool and maybe protects the beer a little bit. I actually don't know, but it's a cool yeast. We barely know anything about it.
Yeah.
Cool. Anything you want to add, Jonah?
No, I mean, it's getting warmer and nicer out there. I know I said fall was the time, but it seems like most of us have nothing but time. So, try and catch something and see if you can make a beverage out of it. I think that's what I'll be doing.
Yeah, no, absolutely, and it's a really great way to get unique flavors out of a local environment.
Yeah.
All right. Well, thank you, Jonah. I think this is super informative. I think we've got a lot of people really interested in this topic, hunting wild yeast and capturing them, and really getting something that is truly their own in brewing. And just do want to mention that for people who hear this and say, "Oh, wow, that sounds like a lot of work." We do also do this for breweries. So if you want to send us a bag of random samples, for a fee, we will process them and find a wild yeast for you.
Yeah.
So that's our commercial plug. Try to keep that limited. And I do want to say, thanks everyone for tuning in. And if you guys are interested in this wild yeast stuff, then we'll try to follow up more and have a little bit more information in the future.
Yeah, totally.
Cool. All right. Thanks, Jonah.
Thank you.
And have a good night, everyone.
Ciao.